Joining online. I can see participants are streaming in, which is great to see. So it's a real privilege to chair and welcome everyone today to our online symposium on updates in COVID-19 in the immunocompromised hosts, hosted by the National Centres for Infection and Cancer at the Peter MacCallum Cancer Centre. So I also begin today by acknowledging the traditional custodians of the land on which we gather today and pay my respects to their elders past, present and emerging. And I extend that respect to any Aboriginal and Torres Strait Islander peoples here today. So I'd like to thank our speakers who I'll introduce shortly and my co-panelists, Drs. Beatrice Sim, Dr. Gemma Reynolds and Prof. Monica Slavin for their help and coordination of today's event. I'd also like to thank Gilead for their on-site sponsorship and refreshments for those attendees tuning in from the Peter Mac site. We will be recording this session, so it will be uploaded to their NCIC website, and we will explore availability via the ACID website as well. So each speaker will discuss their topic for 20 minutes, leaving around 5 to 10 minutes for questions and discussion. So please enter any questions you might have in the Q&A section on this webinar. So without further ado, I will hand over to our first speaker. So it's my pleasure to introduce Dr. Norelle Sherry, who'll be speaking on genomic sequencing of SARS-CoV-2 for surveillance and clinical questions. So Dr. Sherry is a medical microbiologist and ID physician and acting deputy director at the MDU Public Health Laboratory at the Doherty Institute. She's been coordinating the genomic sequencing of SARS-CoV-2 in Victoria since the beginning of the pandemic and co-chairs the National Variants of Concern Working Group of CDGN, which is the Communicable Diseases Genomics Network of Australia, responsible for harmonising genomic methods for surveillance of SARS-CoV-2 across Australia. So I'll stop speaking and hand over to you, Norelle. Thank you.
Great, thanks very much, Toria. Great to see you again, and good morning, everyone. So I guess I'm here with my scientific hat on, doing genome sequencing for SARS-CoV-2, and looking to see what kind of role that plays in the treatment of the immunocompromised host. So just so everyone knows where we're coming from, so I'm working at the MDU Public Health Genomics Laboratory at part of the University of Melbourne at the Doherty, and we provide public health and reference lab services with specialised genomic capability and expertise to support the state and across the region, looking at surveillance, diagnostics and outbreak response. And the main thing is that it's a very multidisciplinary team, and these pre-existing links with both the referral labs, with the genomics capacity and the health departments enabled us to really get into the COVID pandemic pretty much as soon as it came up. So just going back a step, what happens with the diagnostics and sequencing? Well, a patient has a clinical sample, positive PCR, comes to our lab or Vidril, and the viral RNA is amplified and then sequenced. And then we reconstruct the genome from short bits of the DNA sequence, and then align it to a reference, line them all up to see how similar or different they are, and then visualize the relationships between the sequences on a phylogenetic tree. Then we integrate that with the epidemiology and report it. And in Victoria, we've had, I guess, like most jurisdictions, we've had very changing times over the last couple of years. So started with what we thought was biggish waves in 2020. Little did we know, and then a lot of time we had very few samples in 2021 until the third Delta introduction actually finally broke through, and then that morphed into the Omicron wave where we saw massive case numbers that we'd never seen before. And so because of this rapidly changing background, the role of genomics has changed frequently. And I guess the biggest change was variants of concern. So first identified in early 2021 with the Alpha variant. So what is a variant of concern? Well, a variant is actually a really imprecise term, but here it refers to a SARS-CoV-2 virus that differs from the original wild-type virus out of Wuhan. And a variant of concern indicates a group of viruses with evidence of different behavior compared to other circulating viruses. And then there's a range of other kind of levels of alarm, I guess, or levels of concern. So with a VOC being the highest level, and then a VUI, a variant under investigation, and a variant of interest. And those names keep changing, not too much now, but they still do. And then beyond that are other lineages, and lowest level of concern, I guess, is the baseline, the wild type. And the ways that, that a variant of concern, the behaviour differs is either increased viral transmission, increased disease severity, immune evasion, and a part of that is vaccine escape, but also escape from treatments, in particular monoclonal antibodies, and reinfection. So patients are neutralizing antibodies from a previous infection are no longer able to prevent further infection. And so why is that important for your immunocompromised host patients? All very well and good to have this surveillance going on, but what does that mean for doctors and their patients? So each of these VOCs has a unique set of characteristics. And we don't know them when they first turn up. So it's really developing that knowledge on the fly. And sometimes the emerging or the preliminary information that we get is actually a bit misleading, and it's not until probably a couple of months after the wave is over that we actually have a clear view of what is going on or what happened with that variant. So it's really difficult to keep reinventing this wheel in terms of identifying the features of a VOC while we're living through it or even afterwards. So what's important for clinicians is to use this information to discuss risks and prevention measures with your patients. So the role of vaccination, maybe the type of vaccination, although those data aren't usually available until quite a way into the wave, role of pre-exposure prophylaxis, and particularly public health and social measures. So you know, how little do they need to go out? Is it worth them taking the trip up to Queensland, that sort of thing? And it's important to also keep an eye on the treatment options in conjunction with national bodies and guidelines. And so the whole idea behind variant surveillance is to look for a variant that's going to take off, essentially. So that's in terms of transmission. And sometimes we do end up, so we see these different waves. So this was a BA1 wave recently, BA2 wave, and now we've got a combined BA4-5 wave, of which BA5 is the dominant component. So in Victoria, we have a comprehensive statewide VOC surveillance strategy, with genomic surveillance being the key component. So there's a sampling strategy for genomic sequencing of clinical isolates. But this is also supplemented by wastewater sampling and sequencing, and has links with national surveillance and horizon scanning, and in some cases might be supplemented by VOC PCR in surge situations. It really depends on the variant landscape. And so these lineage data in Victoria are being provided back to referring laboratories for their use and analysis. So a lot of hospitals in particular are using that to have a look at the lineages in their patients, but also retrospective views as well. And there's also national genomic surveillance for COVID. So apart from the global platforms, we have a national surveillance platform called AusTracker, where the data are shared privately between public health laboratories with metadata. So that really enables the public health response there. And so that's part of the Communicable Diseases Genomics Network, as well as the National VOC Working Group. So the VOC Working Group meet frequently to make sure that we've got harmonisation of methods and reporting across Australia, but also contribute to that knowledge basis. So a lot of literature reviews, but the way that science is working in COVID is quite different. So it's not just a straightforward literature review. To be honest, it's a a Twitter and preprint review, which is a very different way of working compared to other pathogens. All right, so using genomics to answer clinical questions in COVID. So I've framed this as mostly the immunocompromised host. And so if we think about the immunocompromised host in the middle, there's three main questions that I get. So part of it is outbreak investigations. And so that's a bit of an overlap with the routine surveillance. So it's going from that macro level, the whole population, down to smaller populations. And these are usually healthcare-based outbreaks, looking at healthcare-acquired infections. And the main point of this is to inform infection control policies. So, for example, if there's a patient that would have been cleared by the hospital's internal COVID clearance guidelines, but then appears to be involved, or perhaps the index case of an outbreak on a hematology ward or something like that, it's very important to the hospital to understand whether there was another introduction from outside, or whether it was actually the case clearance policies that might have contributed to an outbreak. And so we do find the immunocompromised patients tend to be included in these outbreak investigations more often than not. And then the other two main questions are for these ICH patients, particularly with prolonged infection or carriage, or sorry, I should say prolonged PCR positivity. So is it actually prolonged infection with the same lineage or strain, or is it reinfection with a different strain? And it can be both. It can be either, actually. And then the other much more difficult question is, what can I use to treat my complex patient? So the phenomena of prolonged PCR positivity has been around well before Vox, actually. So these are data from 2020 that I thought showed it quite nicely. So a minority of cases actually have prolonged PCR positive results more than 40 or 50 days after the initial infection. And actually, even in healthy cases, has been documented up to 120 days. Up to 90 days or more. And some of the difficult part to this is you can actually have negative tests in between. So that probably means that that there's a variation in the viral secretion, but also potentially differences in sampling. And it might just be that the amount of virus in the patient, the viral load, is just around the point where a test can be detected as positive or not. We've also found that something like an intercurrent viral illness, like an ERT, can actually lead to a brief blip again in PCR positivity. But these are usually asymptomatic, these cases of prolonged PCR positivity, particularly in healthy cases. And so prior to Omicron, got lots and lots of these questions. Everyone was on the hunt for the first reinfection case so they could write it up. But the vast majority of these prolonged PCR positive cases were prolonged RNA shedding rather than reinfection. But things have really changed in the last year, I guess, or a bit less than a year. And so this is another study with young asymptomatic adults that aren't immunocompromised. So these are positive results out to 127 days even. And sometimes with that negative PCR result in between the positives, we can actually show that they are genomically identical. So it is actually that prolonged carriage. But we know that immunocompromised hosts have different viral dynamics to normal hosts. So this is just one study that shows the control group. This is in renal transplant patients. Control group, so they have days since admission, and in black the viral load, essentially the log 10 RNA load goes up and then comes down, and there are still several positives, particularly with Omicron, by about 14 to 21 days. But the pattern in the immunocompromised hosts, and these are renal transplant patients, so they're not that immunocompromised generally, often have higher levels of the virus and present for longer. And you can see that in the survival curves here. So there are many case reports and series reporting these prolonged SARS-CoV-2 infection or carriage in immunocompromised hosts. So it is quite complex and there are many factors to consider. So if we think about time along the top, so a patient may have several PCR positives along here and finally a negative at the end. And symptoms could be varied, but usually diminishing over time. So we find that most of the patients with this prolonged carriage might be asymptomatic after a period of time. It's not universally the case. So viral culture is an interesting question, and sometimes that's used to try and understand whether it's just RNA shedding, so shedding of dead virus, or is it actually active replication in the host? And so perhaps that goes for some time. There is a lot of variation here. And then infectivity, so what does that mean? And that's very much an unanswered question. It's something that none of our laboratory measures can easily measure. So maybe infectivity is slightly longer than culture, or it's probably slightly shorter than culture. It's a little bit unclear. And then, so if we do genome sequencing, what are the lineages? Okay, so is it the same lineage all along, therefore it's a prolonged infection? Or is it actually one lineage for the first few samples and then a different lineage for the last ones? And that's reinfection. And we certainly do see both in the immunocompromised cases. So reinfection is I think I would define it as confirmed infection with SARS-CoV-2 after a previous primary infection. And those two infections or two or more infections need to be either separated by time and or laboratory evidence of infection with more than one strain. So either a different lineage, so having Delta and then Omicron, or genomic diversity within a lineage, although this is a lot more difficult to define, but it's something that we can do in limited number of cases. And so the main question is actually how much time needs to go by before something's considered reinfection. And to do that without genomics, so if you're just doing that on a positive PCR, that's a really, really tricky question to answer in the Omicron era. So we used the timeline used to be, okay, it has to be more than 3 months apart. And then, oh, okay, maybe it's more than 2 months apart. And now I think it's down to about 3 or 4 weeks. So those goalposts keep shifting as the VOCs change their behaviour. And so this is an older a sketch of the CDC protocol for investigating suspected reinfection. And you can see the days there. It makes this almost a historical document in the pre-Omicron era. And so one of the difficulties, so ideally we'd have sequencing on everybody and we'd be able to know exactly who had reinfection, but there's now less surveillance altogether. There's less testing of any type. There's certainly rat testing, but probably a lot more symptomatic cases or asymptomatic cases that are being missed. And there's less PCR testing. It's quite selective. And so the numbers of samples are quite large. So it means that with rat testing, subsampling, and limited sample storage of the primary PCR test, it means it's actually unlikely most patients will have more than one sample sequence. So it's very hard to, in a non-defined cohort, to actually, on a population level, to identify how much reinfection there is. But hospital patients may be an exception, so particularly immunocompromised cases. And this is an example of a very early local cases of reinfection where there were 3 cases who were confirmed second wave infections with the D2 strain and then reinfected with Delta. And the reinfection risk really depends on the variant. So there's a lot of data here. It's very hard to summarize, but you can see here significant differences between Alpha, Beta, Delta, and then Omicron. And then there's further differences in Omicron. So BA5, there's a lot more reinfection compared to BA4 and the others. And the reason for this basic difference is there are more mutations, and the particular mutations lead to more immune evasion. And these can be modelled on a molecular basis. these were some interesting reinfection data from South Africa. So they had wild type waves, then beta, delta, and Omicron BA1. And so they noticed a large increase in reinfections in the fourth wave, which is Omicron. And so lineage here is not the only factor, and there's differences between different populations and different locations. So vaccination rates, what vaccination they had, the timing of vaccination, but probably more importantly, previous infections. So what were the rates, and particularly what were the variants? So was it a BA1 wave or a BA2 wave that came before your BA4-5 wave, and the timing. So it is quite complex, and we have to take data from elsewhere with a bit of a grain of salt. So just touching briefly on COVID therapies, so antivirals and antibodies, looking at lots of different complex pathways here. So the usual case where we get asked about this is an ICH case with prolonged infection. So they might be symptomatic or there might not be. Multiple treatments will have already been trialed and they're still PCR positive. The questions will be, is it still the same virus or lineage? That we can answer. What mutations or changes are present in the genome? Also relatively easy to answer. The really tricky part is what treatments can we use? So this is usually the kind of information that we get. There's lots of tests everywhere. There's lots of treatments everywhere. It's very complex. They may have also had some chemotherapy or other treatments for their underlying disease. And then we'll get a number of different mutations that usually come as a stepwise thing, although it's not necessarily a linear thing. So we'll actually see something more like this. So you'll see mutations come and go over the course of treatment. So it is quite complex and very, very hard to interpret what that means. And the genomics here is actually only just part of the picture. So anyone who's been to Dennis Spellman's toots from the Alfred, so it's the organism, the drug, and the patient, or the bug, the drug, and the mug. So with the organism, it's about the lineage, it's about the mutations either within that lineage that are lineage defining or that emerged de novo in the patient. It's also about the viral dynamics, but also about the underlying host. So it's about the immunocompromising conditions, the drugs that are being used, neutralizing antibodies, and vaccination history. And then we have to consider the drugs, so which antivirals, which monoclonals, and what immunomodulatory treatments, as well as the PK/PD. So they all of that information contributes to outcomes for the patients. So how do we use genomic data? How are we starting to use genomic data to inform patient treatment? So ideally we have multiple samples from the patient. We define the lineage and detect the mutations. That's the really easy part. The hard part is reviewing the phenotypes associated with the mutations. So we look at databases and various tools in development. And then the kind of unanswered question is, how do we interpret and report? How do we synthesize these findings? And there's a lot of limitations. So the questions are, how do mutations relate to phenotypes, especially when there's multiple mutations? And some of them can be compensatory and not actually necessarily implicated in resistance. How do phenotypes relate to patient outcomes, particularly in complex patients? And that is something that is really, really, really tricky. And there's the constant introduction of new variants, as I was saying before, means this requires ongoing development. So starting to link mutations and phenotypes, this is the Stanford website here. So there's the two different types of mutations. We're looking at in vitro data here, so fold neutralizing, reduced neutralizing susceptibility in MABs. And yeah, it's tricky to interpret, but we're working on it. And then going from phenotype to treatment. So there's no clinical breakpoints here. It's not like there's a CLSI or UCAST breakpoints for what a fold change in treatment means. So it's a very dynamic space. And we have to bring together data that's phenotypic, maybe inferred phenotype from modeling of binding sites. Ideally clinical data, but we just won't have that data in real time. And the national guidelines attempt to bring some of this information together, but it's a really difficult task with a lot of uncertainty. And then reporting, just briefly, the reports may need to be quite long and complex due to their developmental nature. as really operating outside of our regulatory requirements, and we're not able to validate them. So it could be several pages and likely to be more useful as a discussion. So in conclusion, genomics is another tool in the armamentarium for COVID, has a critical role in surveillance, and that does inform clinicians in terms of modifying preventative measures for patients. But the use in individual cases is less well defined. It's really good to distinguish reinfection from prolonged infection and potentially inform patient treatments, but this is limited by the very incomplete knowledge and constantly changing lineages. Communication of these results can be difficult where the implications are unclear, and I do think a conversation is often better, and so need for ongoing multidisciplinary discussions. And big thanks to all of the diagnostic labs and healthcare facilities that continue to support surveillance, to the MDU individual teams, health department, to my Doherty Institute colleagues, including lots of brilliant virologists and immunologists who are constantly teaching me new things, and the CDGN and National Bulk Working Group. Thanks very much.
Thanks, Norelle. Fantastic talk. So if anyone wants to put any questions in the Q&A, please go ahead. We have a few minutes to discuss. I did have one question. Yeah, you might have a bit more of an idea. Just in terms of the intercurrent ERT and the kind of positivity of the RNA going from negative to positive, that being a trigger. I was just interested what you might think the kind of underlying reason for that might be.
Yeah, it's a good question. I should have put a disclaimer at the front that I'm not a virologist. I think it's kind of analogous to a HIV blip in a patient who's otherwise well controlled. I think there's probably just something in the inflammation in the nasopharynx that means the virus is a bit more available for ends up with more virus on the swab, or maybe there is possibly some more viral replication in there. But yeah, I don't know the answer. I don't think anyone knows for sure.
Yeah, okay, interesting. So we have a question from New Zealand, from Tim Cutfield. Thank you. So he says, thanks for the great talk. Just to clarify, are you making individual patient treatment decisions based on accumulating viral mutation analyses? Are you recommending for or against various COVID-specific therapies like monoclonals versus other antivirals?
Yeah, good question. I think it's a work in progress, and so there's a lot of active work in our national group to provide some of this information with some very heavy caveats to clinicians. So it's certainly not like an E. coli with an S and an R, and yes, go ahead and treat with this, or no, don't treat with that. It's a lot more conditional. It's very preliminary information. It's not at all validated. It's I guess an additional part of clinicians should take these reports when they're finalized as an additional grain of information to use and interpret in the context of the local knowledge, the global knowledge, and national treatment guidelines. I think very, very few things in COVID treatment can be clear and straightforward, particularly when we're talking about new lineages. And I think this is just another piece of information and evidence.
Yeah, fair enough. And kind of in a similar theme, so Prof. Monica Slavens asked, do you have genomic information in regards to recurrent positivity after Paxlovid treatment?
Yes, I actually took that slide out. So rebound after Paxlovid, but actually also other pathogen, other treatments has been noted, and it doesn't seem to be a resistance thing. It seems to be a kind of viral dynamics thing, so a suppression and then reemergence. and it doesn't appear to be resistance within the limitations of incomplete knowledge of the molecular basis of resistance or the genomic basis of resistance for Paxlovid and others.
And yeah, one last question, and then we'll move on to our next speaker. So from Tina Marinelli in Sydney, have you noted any specific mutations developing in immunocompromised patients who have prolonged positivity after receiving Evusheld?
So I think we discussed this on our working group the other day, and I don't think we've had many requests to look for these. There are some mutations that we think are associated with Evusheld resistance, but again, that data is very sparse and just emerging, and it does need to be lineage specific as well. So it's a watch this space. I think if you do see patients who have infections after Evusheld, it would be good to get some of that data together, and that requires just speaking to a local friendly genomics team.
Cool, great. Well, thank you so much, Norelle. That was fantastic. So we'll move on to our next speaker, who I can see in the audience. So thank you for joining us, Dr. Andrew Karaba, who's joining in live all the way from close to Baltimore on the eastern coast of the US. So very honored to have him talk today on SARS-CoV-2 vaccines in solid organ transplant recipients in search of the perfect boost. So Andrew, you can go ahead and try to share your screen if you're able to, and I'll introduce you. So Dr. Andrew Karaba is an assistant professor of medicine in the Division of Infectious Diseases at Johns Hopkins University School of Medicine in Baltimore. He received his BA, MD, and PhD from Northwestern University and completed residency in internal medicine and a fellowship in infectious diseases at Johns Hopkins, where he joined the faculty in 2021. His current research interests include mechanisms of innate immune activation in response to viral infections and innate and adaptive immune responses to viruses in solid organ transplant recipients. So certainly a very topical area. He sees patients as part of the Transplant and Oncology Infectious Disease Consultation Service and is a member of the Johns Hopkins COVID-19 Therapeutic Guidance Committee. So yeah, I can see your slides. So thanks, Andrew. Whenever you're ready, go ahead.
Great, thanks so much, Victoria. You can hear me okay?
Yeah, we can hear you okay, thank you.
Excellent. So I'd like to start off by thanking Victoria and the organizers of this symposium to offer me the opportunity to speak today. I'm really excited to share this work with you. And I'm going to discuss SARS-CoV-2 vaccines in solid organ transplant recipients as a way to sort of frame the vaccines in immunocompromised hosts. And I'll say at the outset, I'm going to discuss a lot of data from different groups. And there's a lot of really wonderful studies that have been done, and I unfortunately don't have the chance to include all of them. So I apologize for that at the outset. So these are my disclosures. And for the talk today, I'm going to first kind of lay the groundwork and show where's our current understanding of the immune response to SARS-CoV-2 vaccines in solid organ transplants recipients. And then I'm going to move on to factors and potential mechanisms that underline a decreased response in this population. And then finally talk about some potential strategies that we might think about employing in the future to improve immunity and protection in this population. So again, we'll start with the groundwork. So I'd like to first start by pointing out that not all immunocompromised populations are equal when it comes to COVID-19 vaccine response. And this is from a very nice paper by Gadi Haidar and his colleagues at the University of Pittsburgh that came out in CID this year, looking at a number of immunosuppressed populations. And as you can see highlighted here, solid organ transplant recipients on average have the lowest seropositivity after vaccination, followed closely to those patients with hematologic malignancy. And so that's why I think talking about this in solid organ transplant recipients is a nice way to frame the discussion for these other populations. So early on in the pandemic, we hypothesized, and then it turned out to be true that solid organ transplant recipients and other immunocompromised hosts would be more susceptible to SARS-CoV-2 and more likely to develop severe COVID-19. And that unfortunately bore out to be true. And as 2021 came around and we started to have the rollout of the vaccines, particularly the mRNA vaccines, my colleagues at Johns Hopkins, this is work led by Brian Boyarsky, Dory Segev, and Dr. Bill Werbel, who you'll hear from later, looked at the antibody response to a cohort of solid organ transplant recipients after 2 doses of mRNA vaccine. And unfortunately, they found that somewhere between 50 and 60% had a positive antibody response after 2 doses. And that's in comparison to, greater than 99% of healthy controls in the study, in the original trials. So this, was concerning that perhaps these vaccines were not going to be as effective in these highly vulnerable groups. And then shortly thereafter, other groups went on to show that the functional immune response was also decreased, both by measuring neutralizing antibodies, as you can see here, and then also looking at the cellular response to the vaccine was also reduced. And as we feared, it turned out that this was predictive of decreased response, or rather decreased vaccine efficacy in solid organ transplant recipients. And solid organ transplant recipients are more likely to develop breakthrough infections that lead to hospitalization and unfortunately even death. This was pointed out by a number of groups, including work done by colleagues of mine at Johns Hopkins. And so because of this, some groups, particularly in France, started to think about giving an extra dose of the mRNA vaccines to solid organ transplant recipients to see if that would improve vaccine efficacy and seroresponse. And work here published by that group in France was an observational study looking at the seroresponse after a 3rd dose. And you can see that a 3rd dose did increase the seropositivity rate and the overall IgG titers to the SARS-CoV-2 spike. And then what was really the premier and definitive study in this area was led by Victoria Hall when she was at the University of Toronto with her colleagues. And they did a randomized placebo-controlled trial of a 3rd dose of the Moderna vaccine in solid organ transplant recipients. And you can see that a 3rd dose did in fact improve the total antibody levels compared to the placebo. And then they also went on to show that this correlated with improved functional response as well, increasing neutralizing antibody titers and CD4 T cells. And so this really led the FDA in the United States and a number of guideline committees to recommend a 3rd dose of vaccine for immunocompromised groups. But even in this really wonderful work, you can see that there's a group of participants, a group of patients that still are not responding to a 3rd dose of vaccine. And so even after this, we were left wondering, how can we improve this? How can we, get this effective for all immunocompromised patients? And so shortly thereafter, an observational study that we conducted in our group at Johns Hopkins, we looked at the vaccine, a 3rd dose of vaccine, and its ability to induce neutralizing antibodies against some variants of concern that were starting to come out. And at the time that we undertook this study, the alpha wave was receding and the delta wave was kind of coming up. And so we compared a group of solid organ transplant recipients in the blue who had received a 3rd dose of vaccine, and these patients had actually gone out and gotten a 3rd dose prior to that being a recommendation. And we compared these to healthy controls that had received the standard 2 dose here in the red. And you can see that while the 3rd dose does improve neutralizing antibodies against these variants of concern, the total antibody response is still decreased compared to healthy controls with the standard 2 dose. And that really is borne out when you look at the healthy controls versus the solid organ transplant recipients and look at the different variants of concern. And we went on to confirm this with a live virus neutralization assay too. So this gave us some hint that perhaps a 3rd dose was not going to cut it as the virus continued to evolve and new variants emerge. And that turned out to be true. If Delta was bad, Omicron was worse. So this was, again, work done by the University of Toronto group, looking at neutralizing antibodies against the vaccine strain, Delta and Omicron, versus after a 3rd dose. And you can see that while there's some reduction in neutralizing with the Delta compared to the vaccine strain, it's significantly worse for Omicron. And so, we think, well, if 3 doses is pretty good and we're losing some efficacy against these new variants, perhaps we need an additional dose. And so in the United States, we were able to do an observational study of solid organ transplant recipients that went out and got a 4th dose, again, before the recommendation. And we looked at neutralizing antibodies against variants of concern. At this time, we were focused really on Omicron. And you can see that while the 4th dose is improving the response to a number of the variants of concern, it really wasn't budging much in Omicron. And while this is perhaps a group that's overrepresented, these data are overrepresented in people who don't respond as well to the vaccine. It's obviously represents a very real group of people that are really not developing a good response, that it would likely be not protective against infection from Omicron. And so this lets us wonder, is there any hope? Can we do anything to help these patients? And I think there is. And one of the things that gave me hope was another study from the University of Toronto that was published quite recently in Nature Communications. And they compared 4 groups of patients. Over here in the pink, they have solid organ transplant recipients that were infected early in the pandemic with non-Omicron strains. And this is a largely unvaccinated group. And then in the orange, they have solid organ transplant recipients that received at least 3 doses of vaccine and then had a breakthrough infection with the original Omicron variant BA1. And then they compared that to, here in the blue, solid organ transplant recipients that had only been vaccinated and not infected. And then as a final comparator, they had vaccinated healthcare workers. And you can see that the solid organ transplant recipients that had a breakthrough infection actually did develop robust neutralizing antibodies against Omicron, actually superior to that of the vaccinated healthcare workers. And they developed robust CD4 T cell responses and even CD8 positive T cell responses, which is encouraging to see because the mRNA vaccines have been previously shown to be less capable of inducing a robust CD8 positive T cell response, even in healthy patients. And so while I don't think anyone would recommend that immunocompromised patients go and get infected after getting vaccinated, at least this shows that there is perhaps a pathway forward and that it might be possible to induce a immune response in these patients. So I'm going to switch gears and talk about some of the factors and potential mechanisms that may be underlying this poor response. And I'm going to start with something that unfortunately is out of our control and out of our patients' control, and that is their prior exposures to other coronaviruses. And so this was an observational study that we did looking at solid organ transplant recipients before and after 2 doses of mRNA vaccine. And we measured antibodies to SARS-CoV-2 spike, but we also measured antibodies against the spike protein of the seasonal coronaviruses. So up here we have HKU1 and OC43, which are beta coronaviruses, similar to SARS-CoV-2. And then the more distantly related alpha coronaviruses, 229E and NL63. And you can see that after 2 doses of vaccine, we do see an increase in SARS-CoV-2 spike antibodies, which is expected. Although again, a number of participants, about half, did not cross the seropositivity threshold. And then if we look at the beta coronaviruses, we see a small but statistically significant increase in antibodies against the seasonal coronaviruses for the beta coronaviruses, but not the alpha. This is a phenomenon known as back boosting, where you have increased antibody responses to related but distinct antigens. And then when we went on to look at, to compare how these pre-existing antibodies correlated with the change in SARS-CoV-2 spike antibodies, here on the x-axis, we have pre-vaccine levels of antibodies against the seasonal coronaviruses. And then on the y-axis, we have the change in the SARS-CoV-2 spike antibodies after vaccination. And you can see that there's a negative correlation such that higher pre-existing antibodies against seasonal coronaviruses are associated with a decreased response to the vaccine. And these associations was statistically significant for all but one of the seasonal coronaviruses. But we do not see such a relationship with an unrelated antigen, the RSV prefusion F. And so we went on to control we went on to adjust for other factors that had been previously published to be associated with a vaccine response in solid organ transplant recipients, including older age being associated with a worse response, receipt of non-liver transplant being associated with a worse response, the use of anti-metabolite immunosuppression being associated with a decreased response, and then closer time to transplant also being associated with a decreased response. And when we adjust for these factors, we still see a significant negative correlation between pre-existing antibodies to seasonal coronaviruses, particularly OC43, and the vaccine and a response to the vaccine. And I think while we can't do anything about people's prior exposures, I think this does raise the question about how effective some of the variant-specific boosters may be in this population. And I think it's an area that we'll need to study as those get rolled out. And I was encouraged to see that they're going to keep the original vaccine strain in the booster. At least that's the current plan, is my understanding. So another factor that we've looked at in our observational cohort is the impact of resting inflammatory state as measured by pro-inflammatory cytokines. And here we took solid organ transplant recipients that received 3 doses of vaccine, and we broke them up into 2 groups: those that developed a high antibody response after a 3rd dose, here in the yellow, and those that developed a low or 0 antibody response after a 3rd dose in the blue, and then compared both of them to healthy healthcare workers in the red. And prior to a 3rd dose, we measured these pro-inflammatory cytokines and saw that a number, even after adjusting for multiple comparisons, were significantly elevated in the solid organ transplant recipients that went on to develop a higher antibody response, suggesting that some low-level reactivity or inflammation, which perhaps is indicative of their level of immunosuppression, is associated with a better response. And then one of the factors that's been pointed out by multiple groups is the use of anti-metabolite immunosuppression and decreased seroresponse, and particularly mycophenolate. And that was seen in the initial Boyarsky study in JAMA published by our group, and then several other groups. And I'm highlighting here a very nice study from a French group, and then a more recent and excellent study by a German group, showing that the use of MMF, and particularly high doses of MMF, is associated with a decreased response to the vaccine. And so we spent some time trying to look into the mechanism as why that might be. And these are data that I recently presented at the American Transplant Congress, and it's work that was really pioneered by Elizabeth Thompson, Andrea Cox, and Justin Bailey as part of our research group. And again, we looked at solid organ transplant recipients that were receiving a 3rd dose of vaccine. And again, there's a number of patients that don't develop a positive response after a 3rd dose, and we call those non-responders here in the pink. And we saw that they were over-enriched, like other groups have, in patients that were on MMF therapy. And so Liz developed a high-dimensional flow cytometry panel to try to characterize their B cells. And she noted that in those that responded to the vaccine, those in the blue color here, green or blue color, had an abundance of these alternative B cells that are marked by CD11C positivity. And she saw that the CD11C positive B cells were spike specific and positively correlated with increasing antibody titers, suggesting that they are contributing significantly to the antibody response. And she also noted that they were upregulating a metabolic marker called CPT1A, which is the rate limiting step in fatty acid metabolism. And so to try to figure out what metabolism or MMF might be impacting these cells and their ability to produce antibodies, she started to do some metabolic assays, both ex vivo directly from patients and then in vitro using PBMCs from healthy patients that she treated with the active metabolite of MMF called MPA. And you can see from these 2 graphs that either patients on high dose MMF already or in PBMCs or B cells treated with high dose MPA, they have decreased mitochondrial respiration, suggesting that that MMF is directly impacting their ability to metabolize fatty acids. And when Liz looked under the microscope at these cells that were treated with high-dose MPA and stained them for lipid droplets, they indeed accumulated lipid droplets, which you can see in this oil red staining here on the left of the slide. And when she blocked the ability to use CPT1A to metabolize fatty acids, in the presence of MPA, you can see that blocking it with this drug atomoxir also decreased the metabolic rate, suggesting that this metabolic function is tied to both MPA and their ability to function and proliferate. And so this leads her to develop this very nice model of how solid organ transplant recipients may respond to the vaccine, even in the presence of T-cell inhibiting therapy. So here in the solid organ transplants recipients that respond to the vaccine, while their T-cell function and number are decreased because of the other immunosuppressants, they're able to access these alternative B cells that need less T-cell help, and they upregulate this metabolic marker CPT1A, and they proliferate and produce antibodies. In the case of the solid organ transplant recipients that are non-responders, they again don't have great T-cell function, and because they're on high-dose MMF, which is inhibiting CPT1A, these alternative B cells can't proliferate, and therefore they are unable to develop a positive antibody response to vaccination. And so finally, the last part of my talk, I'm going to talk about some potential strategies that we might employ in the future to improve the situation for immunocompromised patients. And so we've talked a lot about MMF, and so certainly a number of groups have taken hold of that and done some either observational or uncontrolled studies looking at holding this anti-metabolite immunosuppression prior to receipt of vaccine. And so here's two groups that have shown that holding it from 4 to 7 days prior to a fourth dose of vaccine improves seropositivity and improves neutralization against variants of concern. And I'll plug, some of the work we're doing at Hopkins, where we're currently enrolling a multi-site trial of randomization to immunosuppression reduction prior to vaccination. And so we're currently recruiting patients now and hope to have those data out in the next year. And then going back to work that was done by Victoria Hall when she was at the University of Toronto, this is not in solid organ transplant recipients, but I think it bears mentioning anyway. So in Canada, when they were rolling out the vaccines, they actually, in order to try to get more first doses into people, prolonged the duration between the first dose and the second dose after a period of time. And so they had sort of a non-randomized, but still controlled study where they could look at healthcare workers that had received the standard dosing of 3 weeks versus a more prolonged 8 to 16 week interval. And you can see when they compare the antibody titers in these two groups, those that delayed their second dose actually had higher antibody levels. And so it's possible that altering the dosing schedule in the vaccines may improve the immune response to them. And then finally, I'd like to touch on the possibility of mixing vaccine platforms. And these are data that came out earlier this year by our group. This was led by Teresa Chang and Jen Alejo in our group, and Bill Werbel. And they looked in an observational study at solid organ transplant recipients that either received a third dose of mRNA vaccine or as a third dose received the J&J, which is a adenoviral-based vectored vaccine. And they looked at antibodies 1, 3, and 6 months post third dose, and they noted that those that received the adenoviral vector vaccine actually had an increased odds of seropositivity at 3 months and increased odds of seropositivity and higher antibody levels at 6 months. And so while specifically the J&J vaccine has somewhat fallen out of favor, it's possible that alternative vaccine platforms, either adenoviral-based or protein subunit-based, mixing with the mRNA may provide some improved immunity. And so in summary, I started off by showing you that the immune response to SARS-CoV-2 vaccines among solid organ transplant recipients and other immunosuppressed groups is less robust than healthcare workers, healthy population, and that this effect is more pronounced with the newer variants of concern. And then we discussed a number of factors that may be underlying this decreased response, including the immunosuppressant regimen, particularly the use of MMF, the resting inflammatory state or level of immunosuppression overall, and then exposure to previous coronaviruses, which may be leading to antigenic imprinting and decreasing the ability to respond to vaccination. And then finally, we talked about some possibilities for the future, including immunosuppression reduction prior to vaccination, alternative dosing strategies, and novel vaccine platforms, or just mixing existing vaccines, which may improve protection in these vulnerable groups. And so with that, I'd like to take a moment again to thank Victoria and the organizers of this symposium for the opportunity to talk, and then point out that this, a lot of people, hard work went into some of the studies I discussed, including my colleague Bill Werbel and everyone else at Johns Hopkins. and acknowledge my funding and my lab members, Tidna and Scott, who performed many of the assays. And I'll be happy to take any questions. Thank you.
Thank you, Andrew. That was fantastic and very comprehensive. So yeah, again, if anyone wanted to ask any questions via the Q&A function, please go ahead. Yeah, I have a couple of questions, if that's all right. I'm not sure if the Omicron-specific booster is available yet in the US, but it's definitely looming availability here. And I guess we assume that immunocompromised patients will be one of the priority groups. I guess with your work, thinking about the hybrid immunity that you discussed, so our transplant patients now might have had Omicron or other coronavirus infection or the seasonal coronaviruses, and they've had vaccination against the ancestral virus. So what do you kind of think about the back boosting phenomenon and how this might affect their immune response to the Omicron specific booster, in particular with their immunosuppression as well?
Yeah, I think you're asking excellent questions, Victoria, and I think time will tell, because we're definitely going to study all of those things. I will point out that at least in the U.S., the current plan, as I mentioned, is to have a bivalent vaccine, so like a Omicron sublineage-based version, and then the original ancestral version. And I think that might help some with the antigenic imprinting issue, because it will help boost some of those broader immune responses instead of just focusing just on the Omicron specific antibodies. And then I think what's going to be really interesting is to see, look at patients that had like a Delta infection versus an Omicron infection, and how do they respond differently to the vaccine, and how does that impact the breadth of their antibody response. And then the final, like the moving target, right, or the thing that's continuously moving is time. And so patients that early on may not have responded as well because they were 6 to 12 months post-transplant now are a year or two out. And we know from a lot of good data that the further out from transplant, the further you are away from that induction immunosuppression, the more likely you are to respond. And so that may be helping some of these patients that early on didn't have such a good immune response.
Mm-hmm. Yeah, excellent. All right, so we have a question from the audience. So thanks, Kylie. Yeah, so I think I was wondering about this as well. So in terms of the pausing of the mycophenolate, are you recommending that routinely to patients or currently just in a trial? Yeah, what's the current approach, I guess, at your center?
Yeah, so at Johns Hopkins, we are not recommending doing that prior to vaccination, only in this trial setting. One thing that we found with the trial was, initially when we came up with this, we had a very sort of strict inclusion criteria based on a clinical antibody response. But as time moved on, the goalposts, sorry to use a U.S. sports analogy, but the goalposts have continued to move. And so people are needing higher and higher antibody responses on these clinical assays. And so we've tried to move that to include more participants, because, whereas a level of 500 might have been okay for the original vaccine strain, I don't think that's going to provide the inadequate level of protection. So we are not recommending pausing MMF. There are a couple of centers I've heard about that are doing that, and I know that in patients with rheumatologic illness, where pausing immunosuppression, may not be quite as touchy clinical situation. There are a number of rheumatologists that are recommending pausing prior to vaccination, but I don't think that's a blanket universal recommendation right now.
Mm-hmm.
No, you have gold post in Australia too. Yeah, sorry.
Yeah, and has there been reports of a rejection from, I guess, the kind of mycophenolate holiday and vaccination?
So in those two papers I referenced in my slide today, they did not see any episodes of rejection that were clearly tied to the pause. Now, both of them were small studies. One had about 26 patients and the other one was fewer than 50. But they were not controlled, right? So they were in the second one, where they did have two groups, the decision to hold was sort of based on the transplant physicians. So they sort of were over selecting for people that would probably be fine holding it, maybe didn't even need that extra MMF anymore. And so I think it's hard to say definitively that it's safe to do, but there's growing body of evidence to suggest that it's not causing problems.
Okay, great. All right, thanks, Andrew. So I think we'll wrap up there. Yeah, Tony let us know that we do have goalposts in Australia, which I think provides a good segue to our next speaker. Yeah, so I think Cam has joined. So we'll go next with Dr. Cameron Wolfe, who's been busy.
I can go either way, Tori, if it's easier for Bill or myself, whichever you prefer.
Okay. Yeah, I think it should be okay to go with you, Cam. Yeah, thank you. So Dr. Cameron Wolfe, I will introduce next, who's just switched over to our Zoom from another Zoom, delivering talk about monkeypox, which again is another topical pandemic. So Dr. Cameron Wolfe is currently at Duke University and will discuss navigating COVID-19 in your impending recipient or impending donor. So he's an associate professor of medicine and transplant infectious disease faculty member at Duke University in Durham, North Carolina, an active clinical researcher primarily investigating the impact of viral diseases and their consequences on transplant outcomes, again, very topical. He has a special interest in donor-derived infections and has served as the chair of the UNOS Disease Transmission Advisory Committee and on the UNOS Board of Directors. He also leads the initiative for advancing HIV-positive donor organ transplantation, including serving on the 2014 UNOS Committee and NIH Review Panel for the HOPE Act, which has been fantastic and enables transplanting of HIV-infected organs into HIV-infected patients. So I'll hand over to you, Cam, if you want to go ahead and share your slides.
So I'm on those HIV panels at Duke, to be fair. There are plenty of others who also manage them nationally. So let me share, if I can, bear with me. And maybe if someone can just give me a little thumbs up if that's visible.
Yeah.
Okay. Well, hey, yeah, I do appreciate being a bit schizophrenic, and I've suddenly realized I've jumped into tomorrow on my Zooms. Unfortunately, we're 17 or 18,000 people into a monkeypox outbreak here, including a number of transplant patients now. So apologies for tag teaming into this. So I was asked to give a discussion on what to do with COVID, both if your recipient who's pending a transplant would turn positive, and then also what to do with donors who test positive. And so by means of completion, I've done some DSMB and advisory board work in the COVID space, although none of it impacts this talk at all to my knowledge. So these are the sort of two topics. I'm actually going to talk about looking at donors who are positive first, and then we can move into sort of recipient decisions after that, and certainly happy to take any questions that then come along. This is going to be a talk colored by Duke experience. I will admit and concede from the outset that we may have been more aggressive in our approach, frankly, than some places, and I'll sort of get into why. So I don't want to, for a minute, expect people to feel that this is sort of a national review of the way the US has done COVID. We've done plenty of missteps with COVID, as I'm sure you all know, but this reflects our experience and where we think this is headed. So actually, to take a step back, I'm sure I'm preaching to the converted, and this is, by the way, this is very much a solid organ transplant talk, but this is, we know this is not safe 100% of the time. And it's not safe for a variety of different reasons. You know, we have epidemiology that's changing all the time. West Nile, a great case example. We frequently run into problems that are geographic in their variability, and what affects me here in North Carolina on the East Coast is very different than what affects Australian audiences. You know, we live in a world where the pace of donor evaluation and testing requires window periods and testing sensitivity and specificity that are inherently have some issues. And we live in a world where we often don't get perfect medical history. And so what that plays out is that we sometimes land ourselves in a situation where we're facing infection risk that cannot be mitigated to zero, no matter what we do. Or at least, as we'll get into, in order to mitigate it to zero, transplant programs can't have donors, which is equally an unacceptable and untenable position. Although frankly, it did occur here in the United States for a block of time in March, where I think most transplant programs, at least if didn't stop, substantially reduced their transplant volume. What kind of flipped that whole concept on its head to me, and I think mainly many people from an ID background try and come at the perspective of trying to minimize all risk or seeing an infection transmission as a sort of disappointing event. Actually, the colleagues on here from Hopkins would be well familiar with Dori Segev's sort of paper that was pretty pivotal to me a couple of years ago when I was in the midst of some of my UNOS work, to sort of say, look, occasionally the right thing is actually to say yes. And the reason is that the right thing is to say yes, because sometimes your patients don't get another offer. And it's this was a database review in US data looking at patients with increased intravenous bloodborne pathogen risk, but the premise is the same, that sometimes actually saying no to things that we think are more likely to have infection leaves our recipients fine from that perspective in that immediate moment, but very quickly starts accumulating risk that they're not going to necessarily move to a better organ down the pike. And that is different in the lung program. It looks, actually, we tried to do the same thing with one of our fellows, surgical fellows here, and really showed that this was very similar in that program. In fact, maybe even more urgency in a thoracic space. And I'll get into some of our COVID thoracic data in a minute. But essentially, the more urgent you needed a transplant, the more pressing the decision and sort of urgency to get to a yes answer actually became. And that mortality difference that creeps up, stays with you for really many years. So the way we I'm going to use liver as an example, but we really started a couple of years ago thinking about our waitlist in a management strategy that has evolved over the last couple of years. A very similar strategy actually to the way we look at hepatitis C donors, HIV donors, donors who are mismatched to hepatitis B, in fact, as well now. And so early on in COVID, when we really didn't understand exactly all of the viral dynamics at play, we took a process to say, look, we know our tests are going to be imperfect on donors. We may miss some cases. But we have a wait list, particularly in our thoracic program at Duke, that is heavily skewed, liver too, hence the relevance, that's heavily skewed towards trying to transplant patients who don't actually get onto our wait list until things are critical. So our lung transplant waiting time, for example, is about 14 days once they're activated. The expectation is that they'll get an organ very soon. Liver, a little bit slower. Heart depends a bit on the urgency and where you are in the United States. But the point being that initially in 2020, we took an active decision to say, we have a busy lung program. We do more than 100 lung transplants a year. We will lose more people dying on our wait list if we say no to organs, even if we recognize imperfect testing modalities in the early phase. Other programs have sort of taken a different perspective and have sort of said, maybe I can look at a wait list and take risk into account really early on. These are people who would never usually get organ offers anyway, like low MELD purgatory, or people who are pre-dialysis. We didn't go that way with COVID, although we have with plenty of other donors. Eventually, we all moved, as I'll describe, to a situation that essentially says, if you're sick enough to be on our transplant list, we think you're appropriate for us to consider a COVID positive donor. And we moved into this phase in sort of late, I guess, early 2021. Some groups still sort of taking a little more precise discussion, and I'll get into this in our BAL positive donor lung experience, to say there may be really particularly niche situations where we think someone's going to die on the wait list and we're prepared to take an effort in odd scenarios. And of course, this is a different assessment for patients based on their age, based on their comorbidities, based on many different things that all sort of skew into what do I think's the risk here if I say no for this person. So that's the basis of the sort of framework that I want to set for everyone as to why we did what we did. The other thing that when we take a decision to say, look, we're an aggressive program, we're prepared to take some risks because we think our life expectancy and our waitlist benefits from taking risks, that comes with a recognition that sometimes those risks don't pay off. And I want to start by this because this case framed our conversation here pretty significantly. So we had a woman who had sat on our waitlist for an unusually long time for us, some months, because she had a very difficult PRA mismatch. And came into the organ offer of what seemed like an outstanding pair of lungs, NP swab negative ×2, and has had this, colorful immunosuppression cocktail, should we say, where really this is a sort of novel approach we've been using in rare patients where we either cross major HLAs or have high PRA issues. Unfortunately, 7 days in, she developed really significant pneumonitis and was reintubated. And as I'm sure you are guessing where I'm going, had COVID. We were able to retrospectively find that COVID on a stored BAL sample. Also, ironically, find it in her brother, who was her in-the-room caregiver. At this stage in the US, only one person was allowed in the room. And she actually recovered, maybe because in hindsight, she was getting so much plasmapheresis for her antibodies that we were able to give a lot of convalescent plasma. I don't know. But she's alive and doing well back in her home country now, in fact, 18 months later. And I think whilst I would never want to jinx someone by giving them donor-derived COVID, it was an interesting conclusion that we don't think statistically, given the pace of organ offers she was going to get, that she would be alive at this point had we not been prepared to say yes at that time. Clearly a risk based not only on her individual situation, but also frankly factoring in that we're such a big program that we have some bandwidth to take these sorts of decisions. This was interestingly occurring at exactly the same time in the US when a second case occurred in Toronto and on the back of a third that had already occurred in Michigan. And it led the North American Organ Donation Services to require BAL COVID testing for folks who are going to be lung donors. But it led us to think a little bit more, to go, hang on, how can we protocolize this in a way that makes sense? And so this protocol is what we were doing at the time and continue to do largely for hearts, livers, and kidneys. And this is, I'll start by the outset and saying, this is premised on the, I think, still accurate reality that even though you can find evidence of COVID in tissues outside of the respiratory tree, I don't think there's any supportive evidence to find that you can find replication-capable virus outside of the respiratory tree, at least not that can reactivate in transplant. So we took an approach to say, is there some reason why I might be nervous about organ quality that's COVID specific, i.e., is that person having hypercoagulable disease or hyperinflammatory syndrome markers in some way that I can measure, knowing, again, very imperfect assessments in the sort of acute diagnostic session that you get when you're looking at a donor. But like we had a donor, for example, who had seemingly pretty mild COVID and yet had presented with bilateral PCH arterial strokes. And from a coagulation point of view, we felt that although we were being offered a liver, that this was a little more unpredictable a situation than we would say no. We then proceed to a standard organ offer assessment. Many of the organs that we were receiving early on as a center who was prepared to do this were organs that had come to us from a greater distance, with many centers in the middle that would say no, had come to us as otherwise young, healthy donors where the organ procurement agencies were hoping to make the most of this donation offer. And we would simply say at that point, if they were fine, that we would say yes. We tell our procurement teams to be conscious that the body is clearly potentially infectious, or at least that we should go into a procurement setting using the same PPE that the hospital would require for the management of that individual, but that essentially once the heart or the liver or the kidney was in the box, the tissue was no longer infectious. And that when we returned to Duke, we would treat the person as an uninfected individual. We did not use antivirals in our recipients. We don't use monoclonals in our recipients. We don't manage the infection control setting any differently. Again, presuming the entire premise based on the fact that we think these organs are non-infectious. Our lung program also took this, but recognizing clearly from our own experience, some challenges with infectivity in the lung space, took a more conservative approach and tried to factor in time. So, I'm sure many of you have, particularly in the immunosuppressed world, extensive experience where PCRs can remain positive for weeks, if not months, in some of our patients. But in the general population, that's still uncommon that they would be positive on tests beyond about 20 days. And in particular, we chose 20 as a limitation because that was also the limit to when infection control policy set by the CDC in the US said, look, we don't think there's enough viable virus beyond that point. You can bring your patient out of isolation if they're in the hospital. So we added that here as a surrogate. So if a donor was nasopharyngeal swab positive, but we had a clear history of them having that illness for more than 20 days, we'd head down the same pathway. Nasopharynx positive, BAL negative, we'd use the donor in the lung setting too. Again, similar to what I've mentioned before, this was not a post-exposure setting. We didn't use sort of further antiviral strategies or modified immunosuppression at all because we assumed these were non-infectious individual grafts. To be fair, our assessment of our success here, which I'll get into in a minute, is limited by the fact that we don't routinely look for blood or sort of other invasive measures of looking for COVID, although I will say in retrospect, having gone back and done sort of immunohistochemical analysis of cardiac biopsies, we only saw it in 1 out of 10 cases of an individual who did fine. And we are a routine bronchoscopy center, which includes COVID at least weekly, and we didn't see any transmissions. So our early heart experience, and this was presented at ISHLT by one of our fellows, and to be fair, those dates are censored at the start of April, so you can add a couple of months now to those. The first 11 cases did really well. We had 1 death, but it was the death in someone who was undergoing a heart-liver transplant, having had a history of a Fontana repair in their early years and had some really catastrophic intrathoracic bleeding as a result of previous scarring on day 1. Actually got a second heart from another positive donor, which did well, but he ultimately succumbed of post-surgical issues. Again, not that we think of COVID. So our heart transplant experience actually was really reassuring. And we're now up to about 18 or 20, I think, using BAL or NP positive hearts with really no concern here. Acute or chronic infection doesn't matter to us. Our lung experience, again, presented at ISHLT is a little different. It's clearly shorter. These were patients who were much more carefully screened. Both the cases here had clear histories of having had PCR positivity for a block of time longer, were dying of non-COVID related complications, had all normal parameters in terms of their pulmonary assessments, and have done outstanding for their donors. I will say, off protocol, and I don't have full data on this yet, we do have a number of people who we've now accepted very cautiously who've been BAL positive. And again, they've either been in situations where we knew it was greater than 20 days, or in really unique situations where we thought the testing was different, or that we would lose a recipient in the ensuing day or two if we didn't move forward. And again, knock on wood, these have been fine with no obvious transmissions that we're aware of. I would not for a minute pretend that that's standard of care. But I do think, apologies, my screen's freezing. I do think supports our process now. So when at ATC this year, Lara Danzikov, who currently chairs the committee, looked sort of back at past experience across the country in using these donors. In fact, you can see here that at least organ quality, graft survival, patient survival at 30 days appeared really well matched with taking negative organs, although clearly this is a skewed sample because this is the organs that we thought as a community were good enough to take, not reflecting all COVID positive organs, just the ones that we used. So if folks want to look at how we tipped this, again, Emily Eichenberger, our fellow, presented a lot of this data and really deserves all the credit more than me for doing an outstanding job and putting together a few nice editorials on the topic as well. So our current position, in summary, for donors who are positive, we have we're a transplant center that has a requirement, not a mandate. We can get into the weeds of that ahead of time to pre-vaccinate against multiple things, inclusive of COVID. And we are in a center efficacy unknown, using a lot of Evusheld early post-transplant. Not that that portends any issues here. We cautiously use NP swab positive lung donors, and we'll plan to continue doing that, as I think Omicron is in fact drifting in the way of increasingly mild disease with less lower respiratory tract disease. Very case-by-case BAL positivity, although if you're a liver, heart, kidney, pancreas, or combination of any of the above, I don't really mind at this stage when or where your test was positive. We've talked about intestines. That's the one that gives me some pause. I know of no national programs here who've used COVID positive intestines, and I worry that they may in fact be infectious. We can certainly find PCR positivity on intestinal organs, so we have declined those to this date, although reassuringly they're rare, so I don't think there's a waitlist issue for our fund. So let's flip-flop and talk about recipients who are on the waitlist who test positive. And these kind of come up in a couple of situations, commonly, in fact, in Omicron, and even now in the summer of BA5, we have people who call us and say, look, hey, I've tested positive. Or we still routinely screen everyone who comes in the hospital with an NP swab. And so they may be called in for their transplant and test positive. And we've assumed so far that these are active infections and surgery gets deferred. That's a tough call, because that's potentially a loss of an organ when that comes in, although obviously the US system, and I'm sure in Australia too, have backup patients who usually can make the most of the organ. The deferral length is in flux for us, and I'd love to hear what other people do. We have said a minimum of, originally our minimum was 20 days. And we have been dragging that back progressively as we have found less and less severity in the illness in a heavily vaccinated, and I think much more reassuring variant world. Currently sitting at 14 days and 20, depending on the severity. And really this is based around, both assessments of risk to patients and our staff. We're a big hospital, but in January we were busting at the seams with 1,000 people sick at any one time. But also some early surgical literature that hinted that COVID outcomes were worse. And when you look at some of those papers, I can't find any that are really reflective of Omicron data. They're almost all reflective of, clearly, if you look in the red box and then the green box, some comparatively worse all-coming surgical outcomes. This is a large US 5,500 patient data set that do show early complications due to with the presence of COVID for people undergoing surgery. But this is all pre-vaccination. This is pre-Omicron. And I do think is probably less likely to be as relevant now, although still colors our decision to defer when we can. We certainly defer for living donation, and we make careful decisions about deferring deceased donors. This is kind of currently where we sit. I will concede this is a work in progress. As many things of COVID have evolved, this is kind of our current framework. You can see it's not rigid. If the duration of your positivity is more than 20 days ago, fine, move forward, assuming you're well. If it's less than 20 days and you're in the hospital and I think it's urgent, we've certainly gone much less than that and taken people to surgery. The earliest, I think, is a heart transplant who is our status 1A with a balloon pump, who got nosocomial COVID on our ward, cleared on the back of remdesivir and monoclonal, did not have major symptoms at all, and we transplanted her on day 8 with good success. I know there are programs here in the US who are transplanting as early as day 5. We've not run into that situation, and we certainly don't plan to sort of accelerate or truncate that timeline in our kidney program. The hard part is most of the time you don't know increasingly. Our patients can't tell you exactly when their test was positive, and we sort of approach these cautiously. Again, I think we're probably more cautious than we need to, but recognizing that particularly for kidneys and living donation, this is a semi-elective procedure, so we should be careful. So with that, timely reminder, we like to really vaccinate all of our patients, as I'm sure others in this talk have discussed, pre-transplant if we can, post-transplant in any case. And with that, I'm happy to take questions. I don't know how folk want to run this, but I'll stop the share and take them as we go.
Great, thanks, Cam. Really cutting edge presentation there. So I have a couple of questions in the Q&A, so we might go to them first. So Tina Marinelli from Sydney has commented, for transplant candidates who test positive, currently the TZAN guidelines are to recommend delaying by 7 weeks. But in reality, it's a case-by-case assessment. In Sydney, they've also performed an urgent liver transplant in a patient with acute COVID on day 5. So yeah, I guess that weighing up of risk and benefit, and as you say, we're in a much different kind of scenario now than compared to when some of that data was first released.
Yeah, I mean, we're in a different situation. We're all vaccinated as healthcare workers. We're in a better situation in terms of variant pathogenicity. And I think that sounds like an entirely similar approach to what we would do, which is to assess what's my chance of this patient making it through this time window that I would otherwise rather arbitrarily set. And I think if you're a heart, liver, or lung, you may not have 7 weeks to wait. So I'd roll the dice on that patient any day of the week. Again, recognizing we're a large program with some bandwidth to take, I don't want to say that we sort of gratuitously want to accept hits, but I think the bigger your program is, the more institutionally you can be prepared to do things to get your patients transplanted, that maybe a smaller program does not have the capability of having poor outcomes to tolerate that, if that makes sense. But we would do exactly the same as what Tina described.
Yeah, okay, thanks. And I guess, yeah, a question related to that. So someone has asked, I noted most of your criteria are time-based assessments. Do you utilize CT values in any decision-making? Interested in thoughts.
We try and find them, but ultimately no. And I think there's a couple of reasons. Number one, I think they're just extremely variable. Our OPO community runs many different tests at different times. I have no quality control over who did the OPO swab. I accept that inaccuracy when I stand with a positive in front of me, thinking that it's unlikely to be a false positive in this context, but I cannot evaluate false negatives or false high CT values. So we don't. I will say that, again, off the record, out of publication, some of those BAL positive, NP positive lungs, we have tracked the declining amount of virus as a measure of maybe our own reassurance, but I think that's a really soft science. And I think anyone in the lab would tell you to be super cautious of putting too much weight on CT values. You look at, say, pro sports, and there's great correlation between NP CT values and infectivity on a NFL football field, for example. They can tell you, right, exactly cutoffs of 30, we think you're above that, you're no longer infectious, but they use the same test in the same way at the same time in a very staggered sort of artificial situation, not an organ transplant when I have no idea who ran the test. So we don't use them. Collect them for curiosity, publish them in the reports. They don't bear into our decision making in general. Okay,
thanks. And yeah, I might ask a question. So just interested in the requirement of vaccination, not mandate. There's an interesting kind of ethical debate at ATC, as you would know. And my kind of sub question is just the practice of Evusheld post-transplant. So is this giving it kind of, just before the day of discharge, or what your approach is at Duke?
Yeah, so we give it the day of discharge, sort of here's your jab as you off you go. I'm less convinced by the utility in that. I mean, we're in a situation that may be different for yours in that we have a freely distributed federal supply. We have more than we are using for other situations. I think there's some retained lab efficacy against BA.5, although it's diminished compared to BA.1, and the studies were in Delta. So take them with a grain of salt, but that's what we do. I think your original question was about mandates and requirements, and North Carolina is in a geopolitically purpley red area where we are this little drop of blue amongst an ocean of surrounding red, where the topics of mandates are pretty vitriolically received. And in fact, a neighboring program in South Carolina was hauled in front of their state parliament when they tried to instigate a mandate. So we went no one here would scientifically disagree with the desire to vaccinate all of their candidates pre-transplant. However, we took the approach to align it with all the rest of our vaccine efforts. And in the same way that if you were particularly against, hepatitis B, flu, and your Tdap shot, I would have some difficulties. And we've historically turned some of those people down. There's clear caveats to say, like, if you crash and burn with myocarditis into the ICU today and you've got a balloon pump in your leg and I'm likely to transplant you tomorrow, okay, fine, I may not have time to get you vaccinated. But I do think some of the data about vaccinating before transplant is strong enough to say that you should try and do it if you can. At the end of the day, we've got to be good stewards for the organs.
Yeah. Okay, and last question from Mal in Winnipeg, previous colleague at University of Toronto. So we've seen a few lung transplant candidates that can't clear COVID-19. They have low CT values, but more than 21 days of symptoms. When are they able to be relisted if they can be positive for weeks?
Yeah, great question. So we've only run into that once or twice. We are a transplant center who relists and retransplants, so we get it a little bit. We have basically followed, it's usually a case-by-case discussion. Do I have any measurable activity that there's ongoing inflammation in this individual, yes or no? Do I see a trend line in their CT values in that situation? I think they can be helpful because I have the quality control of how I'm doing the test or who's running them or what platform they're on. We have typically removed their infection control framework in the hospital by day 21, as it sounds like you would do too, and really start the discussion at that point. We have retransplanted people who've been NP-PCR positive, but often in the cases that I can think of have been some months down the road where it's then become clear that they're not going to gain more serviceable lung function in their old lungs. And in that situation, often right before transplant, we'll get another NP swab to use as our new baseline, because frankly, we bronchoscopy people so often that the last thing we want is for their day 3 test to turn positive post second transplant and for everyone to panic about this is reactivated COVID, which we don't think it is. Mm-hmm. Because I think if you put your patients back on infection control barriers in that situation, they get worse care. But that's a great question. I don't.
Great question.
You can sort of see this is a little bit artistic when it comes down to N equals 1 or 2 patients.
Yeah. Okay, great. Well, thank you, Cam. Thanks so much for your time and stepping away from your other monkeypox duties. So we'll let you get back to that if you need to. And
I'll stay on. This will be good fun. Thank you.
Okay, thank you.
Thank you also for people to change their timeline on my behalf.
Oh, no worries. Yeah. So thanks to Dr. William Werbel from Johns Hopkins University. Bill, if you want to start sharing your slides, please go ahead and I'll introduce your topic. So last but not least is Dr. Bill Werbel from Johns Hopkins, who's going to be talking to us today about Evusheld for the prevention of COVID-19 in immunocompromised hosts. So I think now we have a bit of experience with this, but probably can be afforded a bit more from our US colleagues. So Dr. Werbel is a transplant infectious disease clinician scientist interested in personalizing protection against opportunistic infections in immunocompromised persons, particularly following solid organ transplant. He's a current PI of the Johns Hopkins National Prospective Cohort Study of COVID-19 vaccine responses and serves as the Associate Director for Epidemiology and Quantitative Sciences in the Johns Hopkins Transplant Research Center. His training background includes an MD from the University of Michigan at Ann Arbor and an internal medicine residency and chief residency at the Northwestern Feinberg School of Medicine in Chicago. He's a clinical and research fellowship and also performed a clinical and research fellowship in infectious diseases at the Johns Hopkins University in Baltimore and has just completed a PhD in clinical investigation, also with Johns Hopkins. So I'll hand over to you, Bill, for our final talk. Thanks.
Thanks so much, Tori. Thanks, first of all, to you for the invitation, also to the consortium for the opportunity to talk. And some great talks have already been given already. So the topic that I'll be chatting about is the use of Tixagevimab-Cilgavimab, or Evusheld, the injectable therapy for the prevention of COVID-19 in immunocompromised persons. Let's see. These are my funding and disclosures. I think most notably that I've received a speaking fee from AstraZeneca, but the content of this presentation represents my own views and not that of the funders or my employer. So as an outline to orient us, we're going to talk about the concept of monoclonal antibodies broadly, and then this injectable cocktail that I'm mostly going to refer to as T plus C or TNC because it's a very long name. And then we'll talk about some of the neutralization studies that relate to its activity against different variants, and then some of the real-world effectiveness studies that we have. And then lastly, talk about some of the questions that those have given us and some of the future directions. So first, for monoclonal antibody therapy, as many in attendance are familiar, the monoclonals that have been used throughout the pandemic typically target the spike protein, and many target the receptor binding domain, which is where which docks with the ACE2 receptor in human cells and permits infection. They can be single, or they can be combinations or cocktails, like the one we'll be talking about today. And pretty much across the pandemic, as long as an antibody has been used against which a variant is susceptible, there's been a pretty consistent benefit for treatment, particularly early treatment with monoclonals, particularly in people with impaired humoral immunity or lack of humoral immunity, usually measured by anti-spike antibody. And this is just a little screen capture from the recovery preprint that shows almost this concept of like effect modification, meaning that the effect of giving a cocktail, this case was the Regeneron product, was basically not seen in those people who had a prior positive antibody, but there was an effect in a reduction in mortality in people who did not have an antibody prior to its administration, typically early in disease. So we have these tools, but how do we know to whom to target them? And this is a representation of work I helped with in the IDSA produce about just a sort of approach to thinking about who is at highest risk for COVID-19 and severe outcomes, and how we can use that maybe as a proxy for whom we should target. So many, like I said, are familiar that age is still the most powerful risk factor for severe outcomes from COVID-19. Accumulating medical comorbidities also in stepwise fashion contribute. And then vaccination status, obviously people who are fully vaccinated and particularly recently boosted are quite protected. Unvaccinated people are not. And then the most confusing bit and debatable is the concept of immunosuppression and how that impacts the protection that vaccination might afford. And we're mostly going to be talking about people in the red zone here, people who receive lymphodepleting drugs like rituximab, solid organ transplant recipients, and hematological malignancy. And I think what further supports this paradigm for the importance of antibodies is that there is an association between poor antibody response and breakthrough. And it's an association, not necessarily causal, but this is a slide taken from the CDC presentation that helped authorize a third dose in the United States of mRNA vaccine, and showed that the most deeply immunocompromised persons, here in dark blue would be heme malignancy, or in yellow solid organ transplant recipients, have the poorest antibody responses to vaccination, two-dose vaccination. And indeed, in large population studies in the United States, the rates of breakthrough after 2 vaccines was high, and somewhat proportional to this antibody or lack of antibody response, with solid organ transplant recipients represented in the orange here, quickly going away from the rest. And so this is some of this rationale for targeting possibly prophylaxis, and an ounce of prevention is a pound of cure. So we'll talk about what's available now currently for pre-exposure prophylaxis, and it's this combination T and C. It's 2 human anti-RBD binding monoclonal antibodies that were isolated from a convalescent individual, and they were selected for synergy because their binding does not overlap. They don't compete, so you can give them together for synergistic effect. It's also been modified to have a very long half-life, which is why it's dosed in the order of months, and it can be given in multiple routes. The trials that helped authorize it were mainly this PROVENT trial, which is a phase 3 randomized study at multiple sites of the 150/150 dose. I'm going to refer to dosing in different ways throughout here to sort of do the sort of low and high dose, if you will. So this is the sort of low dose that was designed for people who would be at high risk for disease, harkening back to that continuum slide I showed, including potentially immunocompromised people, but they comprised only 1% of all people in this study of thousands of people. Again, it was also in the pre-Omicron era and in unvaccinated individuals. So very different than where we're trying to use it now. Regardless, though, the results were encouraging in that even though symptomatic COVID-19 was uncommon, there was a relative risk reduction of almost 80% in the people who received the drug versus who did not. And there were no cases of severe COVID-19 or death. So this was the grounds for its authorization. I made a little note here that for asymptomatic COVID, the efficacy was lower, not surprisingly, but relevant for the group that we're talking about, where something that's asymptomatic or post-asymptomatic in an average person may be more severe in a vaccinated immunocompromised person. So there is a lot of equipoise about using the drug, and I want to talk through a little bit. In the United States, it's authorized, it's recommended at this higher dose, the 300/300 dose. But in the UK, very recently, they said, we're not buying Evusheld. There's not enough data for it. And similarly in Canada, other part of the Commonwealth, it's not recommended currently. These are as of this month. So the reason why is related to the Omicron variant, basically. And this is a really nice sketch from a Nature article that shows the distance that Omicron is from the rest of the tree, and then how BA4 and 5 have sort of sprung off of BA2, most likely, and have accumulated many, many mutations in the spike protein, which is obviously problematic because monoclonal antibodies, their strength and their weakness is that they're targeted to a specific area of spike protein. If that mutates, they may not be effective. And in the US right now, BA5 dominates in the sort of teal color, and BA4.6 that we'll briefly talk about is slowly rising. And all of these sublineages, subvariants, can significantly affect neutralization. So we'll talk for the next 5 or so minutes about neutralization, maybe 10 minutes, and then effectiveness. So things that I would encourage if you're looking at neutralization studies are to consider whether the study you're seeing is in people or in a test tube. A lot of these are kind of concentration antibody studies versus those from plasma samples. Particularly, we would care about ideally immunocompromised and vaccinated people. We need to obviously pay attention to the sublineages of Omicron that are tested, and then the nature of how the testing and the neutralization is assessed. Live virus is the gold standard, but not high throughput. So they're mostly done in surrogate neutralization settings. So we'll go through a few of what's available. This is in persons with autoimmune disease, particularly those who received anti-CD20 or rituximab, who have very poor antibody responses. So it's about 30 people who received that low dose T and C, and compared their neutralizing activity at sort of peak level after injection. And if you look on the right here, the gray dots are neutralization against Delta, which is quite high levels of neutralizing antibody, versus Omicron BA.1, similar to the original Omicron, there was minimal, if any, effectiveness, or immunogenicity rather, whereas BA.2, the next wave, showed moderate degree of activity. And you can see the fold reduction here and what that might entail, or what that might imply rather. BA.1 not effective, BA.2 potentially effective. So this is an autoimmune disease. In persons with cancer, very similar. Small studies. These are people, however, who now they were testing the impact of the increased dose, which is currently recommended in the United States, and looking at this sort of hodgepodge of people with hematological malignancies, including quite a number who had stem cell transplants, and many of whom had 3 vaccines prior to receiving the pre-exposure prophylaxis. And then as a surrogate measure of neutralization against Omicron BA.1, which we were worried about in the first slide, obviously, there was at peak level some neutralization achieved in the 300/300 group, albeit not extremely high, but higher than in the half dose group, which you might kind of appreciate. Now, the BA.4.5, which is what we are mostly caring about, as you see, those are the variants or the sublineages that are globally predominant. There have been a couple of studies that we'll share, including some that are either not peer-reviewed or unpublished. The first is a paper that some of our colleagues collaborated with in the New England Journal, and these are one of those monoclonal titrating studies. So in vitro, totally studied. It looked at a slew of different antibodies and antivirals, and across in the rows here, the different Omicron lineages. And I really just, for the purposes of this talk, focus on the red boxes, which are looking at BA.4 and 5, that showed that there's essentially no effect, no neutralization, even at peak sort of undiluted levels for Tixagevimab, one of the monoclonals in the cocktail, but some activity for the other component. And when they were combined together, BA.4 showed reasonable neutralizing activity and a little bit worse against BA.5, moderately worse against BA.5, not just a little bit worse, but better than against the BA.1 lineages that we are the first waves that we are that are predicted to have poor sensitivity. And this is work led by Andrew Karaba, who you just heard about, that's currently unpublished, looking at solid organ transplant recipients and using the higher dose that's recommended in the US in fully vaccinated organ transplant recipients using live virus assays. And we showed here on this plot that against the wild type on the left, or ancestral rather, extremely good neutralization at 2 weeks after injection. BA.1.1 quite poor, with half of the people showing no neutralizing activity. BA.2.12.1, which is previously predominant, now BA.4, which shows a spike protein with BA.5. Again, moderate level, tenfold worse at least than the ancestral variant, but better than BA.1.1. So some signs of neutralization at peak level of the higher dose in immunocompromised persons, fully vaccinated. So as sort of a summary here, because this is, I think, one of the reasons why this equipoise is that the BA.1 lineages, the first waves, are not predicted to have been prevented really by people who received T and C, whereas BA.2, as we saw in the one study, shows better neutralizing activity. And now we're in this sort of yellow zone with accumulating mutations, some of which have been bolded here, that interfere with some of the binding of the monoclonals, but there may be some activity. And I'm just, for comparison, showing Sotrovimab, which was one of the first earlier Omicron active antiviral monoclonal antibodies that we thought we could potentially use, which then lost activity fully against BA.2 through 5. And Bebtelovimab, which we use for treatment in the United States, does maintain activity against all sublineages. So for now. Okay, the next two slides, last two slides of neutralization, I'll just talk about durability, because we talked a little bit about the impact of dose, but how long does it last? And this top graph is from the Provent publication, looking at the sort of peak and wane of the drug level over time, which, as you see, peaks at somewhere between 2 and 4 weeks, then wanes down to about 90 days, and then continues down to day 180. What was done by a French group in the lower half of this slide was looking at antibody levels, the binding antibody unit, and looking at the blue line, kind of seeing a similar trend in these fully vaccinated kidney transplant recipients, where you can sort of see it peaking at 3-4 weeks, then sort of settling down and flattening at about 90 days. And they did show in this next connected line plot that there was neutralizing antibody against BA.2, even 4 to 5 months after injection, but it did wane. And remember, this is the sublineage that we think there'd be the most activity. So BA.4.5 would be predicted to be worse. And in fact, at Johns Hopkins, again, work that's led by Dr. Karaba and myself, in our national observational cohort of vaccinated organ transplant recipients, we did follow a group of people who received the full dose of T and C who were fully vaccinated in serial plasma sampling pre and post the injection to look at surrogate neutralization of BA.5. And what we showed in here is this connected line plot from baseline to 2 weeks, which is near peak level, to 3 months, that the majority of people wane. The majority of people showed activity on this assay by 2 weeks, but around 2/3 to 75% showed waning below the level that we would be, we've shown to be associated with neutralizing antibody by 3 months. And this is the only other study I could find on duration neutralization, which showed relatively similar things. This is from another group that looked at people with autoimmune disease. And most of these people had received a lower dose. And again, as a surrogate neutralization method, if you look at the graph on the right, kind of showing a rapid peak in the first couple of weeks, and that sort of plateaus by day 90 and then sort of plateaued and then went waned after between day 90 and 180. So that was the neutralization data, which we'll revisit at the end, but you can see how it varies a lot by sublineage tested and dose. Now we'll look at effectiveness, and these are some considerations that I think we need to continue to apply when we look at these data. Number one is dose, like I mentioned. Number two is who is receiving these medications and what is their immune substrate, which wave, and what is the follow-up time of people who are being followed after injection that might suggest changes or might have impact on the effectiveness of the drug, and then how are the COVID-19 diagnoses defined and ascertained, asymptomatic, severe disease, etc. And lastly, ideally you'd have matching in some way, but that's actually very difficult in real time during a pandemic. So the first real world effectiveness data is out of France. Much of this is out of France, which was in kidney transplant recipients, many of whom were fully vaccinated and all were shown to have low or nil antibody prior to being given the lower dose, the 150 dose. And these were in the majority BA1 wave. So you can already see how this may have been an issue. And out of the people they followed, 10% were infected at a median of a couple of weeks after injection. So quite early, maybe not when the drug had achieved full capacity. And there were 14 hospitalizations of these. So almost 40% were hospitalized, and there were 2 deaths. So their conclusion, which I would agree with, which is low dose of the Evusheld in the BA1 era was not effective in protecting high-risk organ transplant recipients. This group followed the same people longer as it stretched into the BA2 wave and tried to find comparators as best they could, and noted that the incidence of COVID-19 in the people who received the injection, the TNC, did decrease significantly as they moved out to February and March after the BA1 wave. And in an unmatched comparison showed that there was a significant difference in the incidence of COVID-19 in people who in the red line were unprotected versus people in the blue line who had received this immunoprophylaxis. So they concluded that there may be some effect, but as you can see, this shows this impact of follow-up time in predominant sublineage. I think one of the best studies that I think we've seen to date was out of MGH, which followed over 200 solid organ transplant recipients, almost all of whom were fully vaccinated, and more than half had received the higher dose. And they found a control as best they could based upon how many the number of vaccines that an individual had received. Followed them for about 3 months and noted, as you can see in these cumulative incidence plots on the right, that sort of no matter how they sliced it, there was less, there were fewer diagnoses of COVID-19 in people who received the TNC prophylaxis. In fact, it was only about a third number of infections. There was only 1 hospitalization and no deaths in the people who received the prophylaxis. Notably, the breakthroughs were either at only a couple of weeks or over 90 days. So, and most of the breakthroughs were in people who received lower dose. So in summary, and the higher dose in particular, as you can, that's actually represented in the bottom plot of these 3 super lined up curves. The higher dose seemed to be effective against COVID-19 in solid organ transplant recipients, at least up to around 90 days, where you start to see an uptick, at least in the lower dose in terms of infections. And the last study, which is a little more complicated, was a very large study out of Israel, 5,000 immunocompromised people in this health center, or a real smattering of different kinds of immunocompromise, most of whom received full vaccination, and over 800 of which had received a lower dose, primarily during the BA2 era. And they showed, and I'll show the Kaplan-Meier curve, or the hazard ratio curves here, that they showed about a 50% reduction, again, in a BA2 predominant era, of the people who received the T plus C versus those who did not. And what was a little more hard to explain was that there was a vanishingly small amount of severe disease in the people who received Evusheld versus those who did not, which probably suggests some confounding built into how these 2 cohorts were assembled, but they did note that As you see in the bottom plot here, there were basically no deaths or severe hospitalizations in the people who received Evusheld versus those who did not. So I think a grain of salt with this, but in keeping with the theme we've discussed. So I want to just summarize that because there's a lot of information. I think these data support that neutralization against BA1 of really probably either dose of Evusheld is unlikely to be effective, generally poor. Neutralization is better against BA2, and even low dose seems to show some real world effectiveness. There's probably at least intermediate evasion by BA4 and 5 based upon the neutralization data, but we don't have great real world effectiveness yet. And in general, though, the durability, as we showed in some of those line plots from our group, likely tails off by around 90 days, even with full dose. The last piece of actual data to show, is about safety and tolerability, which I think is always part of the risk benefit calculus. And the thing that has made the most news is that there was an imbalance in cardiovascular events in the ProVed trial and in the FDA documentation of about threefold higher rate. These were all in individuals who had prior cardiovascular disease, and these were a smattering: heart failure, heart attack, tachyarrhythmia. And so this has been an ongoing open question. There was also one case of anaphylaxis, and we've all seen some rate of anaphylaxis with monoclonal antibodies in general. I will say that in work that we have, this is in a preprint, that the drug is relatively well tolerated. That's these bar graphs I'm showing. Slightly more local discomfort with the higher dose injection and some more mild fatigue, but no severe events, at least in the small cohort that we followed. We have a larger cohort of people who've received Evusheld, most of whom received the higher dose. It's over 200 organ transplant recipients at Hopkins, and we only had 5 episodes of cardiovascular events, one of which occurred was an episode of atrial flutter within 4 days of the injection, but the rest were over 6 weeks later. None of them were myocardial infarction or heart failure, so we felt in this with limitations not likely related to drug. And then lastly, in many organ transplant studies that have been presented, there were not reports of rejection or kidney or liver dysfunction. All right, and so the last key questions that I think this gave us is, what's next? We've seen that the Omicron variant changes rapidly, and there are several sublineages that are emerging, like BA2.75 in India on the backbone of Delta infection evasion, and BA4.6 that I pointed out is rising in the United States. And this is a preprint from just a couple of weeks ago on the right that is sort of a heat map. If you look at the second to bottom row, even though there's this reasonable effect, inhibitory effect of Evusheld in the BA4/5, as you start to add even small mutations such as BA4.6 or version at position 346, you can basically lose activity, at least in the dish. I think the data also show that we don't necessarily totally know the optimal dose and durability and redosing schedule here for the drug. Why this is relevant is that, for example, in the FDA in June said, oh, we should redose based upon modeling studies every 6 months, but I'm not so sure in the BA4/5 era that necessarily is optimal. So this will have to be studied. And then lastly, and this was mentioned in one of the earlier talks, is what about the threat of emergent resistance under the cover of MAB, so to speak? A chronically infected individual with sort of suboptimal activity may lead to really problematic mutations. And then even though I touched on some of the real world data of cardiovascular disease, it's a small number. We need to ensure that in certain populations who have high risk, like solid organ transplant recipients, that indeed there's not really a signal we need to be worried about. So with that, I want to thank first my research group at Hopkins. Is really a litany of people who have a lot of smiling faces here, who make the cohort run, which is now about 8,000 people. So thanks for that. And then we also have a legion of scientists also and collaborators that I admire, including Dr. Karaba, who gave a talk today, who help us make our trials move forward. And then lastly, our research center at Hopkins, and then our collaborators at NYU, led by Dori Segev, who was my mentor, one of my mentors, and they do great work. So we're looking forward to contributing to the science as we go forward. So thanks for your attention.
Thanks, Bill. Fantastic talk and a good update for us. So I'll just let you know, in Australia, Evusheld is licensed at the 150 dose. So I guess we're kind of interested and we'll be watching our cohorts as well in terms of breakthrough infection frequency and severity. So I have a couple of questions. So we saw that uptick in the AJT paper by the MGH study group in the 150 group at 90 days. So just interested in your thoughts of redosing. Should we be redosing earlier than 6 months? Is anyone doing that in the US? What are you doing at Johns Hopkins? What are the thoughts?
Yeah, I mean, we are bound because EUA criteria are very strict. I mean, we cannot stray from what's recommended. So we can't do that. I'm concerned based upon these neutralization data that particularly in high-risk people, I think 6 months is probably going to leave a tail of risk. And 3 months might fit a little bit better with what's been published so far. That's a bit of a hard thing to operationalize. It's hard to get people in for one of these injections, let alone 4 times a year. And I think we probably just need to see what happens with the new subvariants and things too, because if you put in all the effort to make that kind of program and then we have a BA4.6 predominance and the drug's not effective, predicted, we don't know. I would be that, that's may not be. We have narrow windows to make these drugs work. And I think we just try to support getting as many people their first full doses that we can, and then keep tabs on them and don't we do not give them sort of false assurance. We say like that's something to augment your protection in addition to vaccination. You need to continue to get boosted on schedule, which is right now every 3 or 4 months in the US for these people. So we're leaning on that as best we can, plus encouraging pharmacological interventions like avoiding crowds, masking, etc.
Yeah, and Prof. Monica Slavens asked, do you have any data for using Evusheld as treatment? So I guess in patients who are coming in with COVID-19 or prolonged shedders, have you been using it as a treatment?
So I will say that the TACKLE study, which is in, I think, Lancet Respiratory Medicine, did test the intravenous drug as early treatment that showed an effect. But for Evusheld, the post-exposure prophylaxis study called Storm Chaser failed. And it's giving intramuscular injection. It takes several days to come up. By that time, you may have missed a window for a monoclonal. So we don't use it for treatment. There is one case with which Andrew is familiar where it had been given along the way, so to speak, in someone's infection, and it was not effective. So we have not been doing that. But we also are fortunate that we have access to bebtelovimab, which is highly active, and really we've been leaning on that for high-risk people.
And so in patients who have had Evusheld with breakthrough infection, what is your go-to? Is it antiviral or is it bebtelovimab? We don't have that available, but interested.
If we have bebtelovimab, we really do try to get that to them, plus remdesivir.
Right.
We run into trouble, as many people have, obviously, when you give Paxlovid due to drug-drug interactions for high-risk people, and the data for effectiveness of molnupiravir is not that exciting. We have done it, but we try to get people in for the infusion center to get bebtelovimab and remdesivir, and continue to really lean on that.
Okay, cool. So Tony Common has asked, thanks for your informative presentation. Can you discuss COVID vaccination and MAB therapy timing? So I guess pre-Evusheld or other MAB, and then post, when are you vaccinating against COVID?
Right. So we tend to fall back on, particularly with these variants and uncertainty, on FDA recommendations, which are, wait 14 days, at least, space a monoclonal vaccination 14 days after monoclonal. There is only one study I know of that's looked at the impact of monoclonal on vaccination titers, which is in Science Translational Medicine with bamlanivimab, which showed about a 2-fold reduction in binding antibody in the people who received bamlanivimab, which was the take of the article was, this is not thought to be really interfering, but in people who might only have a little bit of antibody, it might be a problem. So I do think about that, but in general, we have more faith in the vaccines, and we consider the monoclonals as just add-ons. So we try to get them as vaccinated as we can, and I generally have, it's very case by case, but many times I say, please get vaccinated if you're due for a booster, wait a little bit, then have Evusheld. And if you've already had Evusheld, we'll wait a little bit and then you can get vaccinated. These are usually in the order of weeks.
Okay, thanks. And I think you've kind of touched on this a bit. So from Tim Cupfield, do you think MAB-based strategies are likely to have longevity as we head into the next phases of the pandemic in light of the rate of viral evolution?
Yeah, unfortunately, uncertain. We've seen that with the changing variants that we have these short shelf lives. And so I do encourage the use because we have these windows. And so when we have it, we try to use it. But as I mentioned earlier, the strength and the weakness is if things are very targeted, small changes can lead to loss. And so I don't think any one monoclonal is likely to be forever, is to be evergreen. We'll just have to sort of keep up. And genomic surveillance can be very informative, actually. It's just something that's hard to operationalize.
Yeah, okay, great. So I think we'll wrap up there. So thanks so much, Bill, and thanks to our other speakers, Cam and Andrew and Norelle. Fantastic talks and really informative and comprehensive. We will have this recorded, I mentioned at the start, so it will be uploaded to our NCIC website, and we'll look to upload it to ACID as well. So I'll end there, and thanks everyone for your participation and attendance. Thank you.
Thanks, Tori.
Thanks.
Great talk, Bill.
Thanks, Kate.